Hi, so we will continue with the question number 1. The question number 1 is asked to identify and describe the N-terminal and C-terminal of this peptide. So, a peptide has two ends, which the end with a free amino acid group, N-terminal, this is N-terminal, is called N-terminal amino acid residue. This is amide NH3. And the end with a free carboxyl group, which is the C-terminal, is amino acid residue. This is C-terminal, which is end with free carboxyl group COOH. So continue to the question number 2. The question number 2 is asked to show and explain the peptide linkage. Peptide linkage, also known as peptide bond. So let's see with our peptide. The peptide linkage is an amide formed between COOH and NH2 group by elimination of water molecule. It's represented as COH and NH2. There is 4 amide formed in this peptide, so there are 4 peptide linkage. This is the first one, the second one, the third one, and this is the fourth one. So it is CO combined with the NH2. Next, the question number 3. The question number 3 is asked to show where disulfide bridge likely to happen and explain the importance. So in this peptide, the disulfide bridge is this one, when the sulfide molecule combine with the sulfide molecule form the disulfide bridge. So the importance of disulfide bridge is to enhance the conformational stability of a protein by decreasing flexibility and entropy of unfolded state. The disulfide bonds also function to stabilize the tertiary or quaternary structures of proteins and may be intraprotein, which stabilizing the folding of single polypeptide chain, or interprotein, which is the multi-subunit protein such as antibodies. Then the last question is asked to show where the hydrogen bond likely to happen and explain its importance. Okay, based on this structure, there are 2 hydrogen bonds formed in this peptide. The first one is this hydrogen meet with this oxygen. Then the second one is this oxygen meet with this hydrogen, form 2 hydrogen bond. Not to forget the last question, which asks, is there any difference between this structure, which is CVSAGC, with the new structure which is CGASVC? And the question also asks to show the difference between these 2 peptides. Okay, so this is the first structure, which is CVCAGC. For this structure, we have one disulfide bridge. This is sulfide, so disulfide bridge. The second one is 2 hydrogen bond. Hydrogen bond is when when oxygen molecule meet with the hydrogen molecule, so it's formed hydrogen bond. And in this peptide, there are 2 hydrogen bond, 2 hydrogen bond formed, and the third one is peptide linkage. Peptide linkage is when the NH2 meet with the COOH, so it's form peptide linkage. There are 4 peptide linkage formed in this peptide. So this is the second structure. Excuse for the purple molecule supposed to replace with the yellow, which is represent the sulfide molecule, but there is not enough of yellow ball, so we use purple ball to symbolize as sulfide. So also excuse the trigonal nitrogen. It's supposed to have divalent nitrogen, but there is also not enough. So for this structure, which is CGASVC, for this peptide, there are 1 disulfide bridge, which disulfide molecule will combine to form the disulfide bridge. Then 2 hydrogen bond form, which is oxygen molecule meet with the hydrogen molecule. And in this peptide, there are 5 peptide linkage form. Peptide linkage is when the COOH meet with the NH2 meet with COOH, form the peptide linkage. There are 5 peptide linkage in this molecule, in this peptide. So the difference between first peptide and the second peptide is the first peptide have 4 peptide linkage, and the second peptide have more peptide linkage, which is 5 peptide linkage. So that's it.
And question 2 is, it says build ribose and deoxyribose sugars. So my first question is question A. It says describe using my model marker of C2-endo and C3-endo for each. So here we go. C2-endo and C3-endo are terms used to describe the orientation of substituent groups on cyclic molecules with 5 or 6 membered rings. In C2-endo conformation, the substituent group is oriented towards the inside of the ring and is located on the same face as the bridgehead carbon atom C2. This conformation is commonly found in cyclic molecules such as steroids and sugars. In C3-endo conformation, the substituent group is also oriented towards the inside of the ring but is located on the opposite face of the bridgehead carbon atom C3, as compared to the C2-endo conformation. This conformation is commonly found in cyclic molecules such as pyrenosis and furanosis. The puckering of a ring in either C2-endo or C3-endo conformation depends on the number of atoms in the ring and the nature of the substituent groups attached to it. Generally, a cyclic molecule in the C2-endo conformation will have a slightly puckered ring, while a cyclic molecule in the C3-endo conformation will have a more flattened ring. The exact nature and degree of puckering, however, can vary greatly depending on the specific molecule and its environment. Various computational and experimental methods are used to study the puckering behavior of cyclic molecules. And there are conformations which can have significant implications for their biological and chemical properties. Question B is, it says, can a uracil connected to these sugars, ribose and deoxyribose? So the answer is uracil is a molecule that can be connected to sugar molecules to form nucleosides. Nucleosides are composed of a sugar molecule and a nitrogenous base like uracil. Depending on the sugar molecule, the resulting nucleoside is called either uridine or deoxyuridine. These nucleosides can have phosphate groups added to them to form nucleotides, which are the building blocks of RNA and DNA. Uracil-containing nucleotides and nucleosides are important in many biological processes and can also be used in medicine to treat certain diseases. Last question, it says, I have to build a nucleotide. So I will show you in the next video. A nucleotide consists of 3 molecules. First is a nitrogenous base, second a 5-carbon sugar, and the third one is a phosphate group. The nitrogenous base can be adenine, which is A, cytosine, C, guanine, G, or thymine, T, in DNA, or uracil, U, in RNA. The 5-carbon sugar can be ribose in RNA or deoxyribose in DNA. And the phosphate group is a phosphoric acid molecule that is attached to the sugar molecule. Together, these 3 molecules form a nucleotide, which is the basic building block of nucleic acids such as DNA and RNA.
For question number 3, build alpha D glucose and beta D fructose cyclic structure. So this is alpha D glucose. So this is beta D fructose. Next question is A. Describe beta D glucose structure. Beta D glucose is a monosaccharide, which means it is a single sugar molecule that cannot be broken down into simple sugar. Its chemical formula is C6H12NO6 and known as a hexosis. The beta in beta D glucose refer to the orientation of the hydroxyl group attached for the first carbon atom in the molecule. The OH group is located on the same side as the CH2OH group attached to the 5th carbon. The structure of beta-D-glucose is a 6-membered ring called pyranose ring, formed by the reaction of the hydroxyl group on the 5th carbon atom with the carbonyl group on the first carbon. The resulting ring structure has 5 carbon atoms and 1 oxygen, and remaining carbon and oxygen atoms extend from the ring as a functional group. Next question is B. For B, I need to form a maltose and show where is the glycosidic bond NME. So this is a maltose structure. This is a glycosidic acid. As we know, maltose consists of 2 molecules of glucose that are linked by alpha 1,4 glycosidic bond. This question is C. Explain why maltose is reducing sugar. Why maltose is reducing sugar? Maltose is reducing sugar because it has 3 aldehyde groups CHO in structure, which can undergo oxidation reaction. In maltose, the aldehyde group is located on the first glucose molecule, which is linked to the second glucose molecule via glycosidic bond. The aldehyde group in first glucose molecule can react with kinetic reagent or tolerance reagent, which are commonly used to test for reducing sugar. During the reaction, the aldehyde group is oxidized to carboxylic acid, while kinetic or tolerant reagent is reduced, resulting in Carder chain or the formation of silver mirror. Maltose can broken down to glucose by the maltose enzyme, which catalyzes the hydrolysis of the glycosidic bond.
Good evening everyone, myself Yashwanta. So today I'm gonna explain on several questions. So let's get into the first question, which is which of the substrate above preferred by urease for catalyze.
So this is the structure of urea. Urease is an enzyme that catalyzes the hydrolysis of urea to produce ammonia and carbon dioxide. So the enzyme is specific for urea as a substrate and does not have any significant activity with other substrates. So urea binds specifically to urea through a bind site of enzyme surface where it initiates the hydrolysis reaction.
So there is also a central strain model which suggests that the enzyme creates strain on the substrate to lower the activation energy required for the reaction to occur. So this model proposed that the enzyme and the substrate have complementary shape prior the binding, but the enzyme applies force or strain on substance to facilitate the reaction. So for the second question, explain the model or the hypothesis that describe the action of this enzyme on the substrate.
So basically for the second question, it is the lock and key hypothesis. So which proposed the active site of enzyme is exactly complementary to the shape of the substrate. So the random movement of urease, which is the enzyme, and the urea, the substrate, which bring urea, which represent the key, binds to the active site of urease, which represent the lock. So urea binds temporarily to the active site of urease through the weak bond to form enzyme substrate molecule. So the formation of complex lowers the activation energy and make it easier for the urea, the substrate, to change into carbon dioxide and ammonia carbonate. The product with the different shape has lower affinity for the active site, and it releases from the enzyme, which returns to the original conformation. So overall, the specific model hypothesis that describes enzyme-substrate interaction may depend on the specific enzyme and substrate involved as well as the condition of the reaction.
So for the last question, which is there any other hypothesis for the enzyme to the substrate interaction?
So the third question, the answer is yes. There are various hypotheses and models that describe the interaction between enzymes and their substrate. Another model is the lock and key model, which proposes that the enzyme and the substrate have a complementary shape that allows them to fix together like the lock and key. In this model, the enzyme substrate are true to have specific pre-existing shape that enable them to bind one another.